Hello, my name is Dr. Roderick L. Roll, and today I will be talking to you about proteins. This chart shows you how much protein you are supposed to consume per day, and your consumption is based on your body weight. So 16% of your caloric intake should be proteins. Now, how to figure that out, you need to take your body weight and convert it into kilograms, and then divide that by 0.8. That will give you the recommended amount of protein that you need to consume for your body size. Now, if you consume too much protein, listed below are a few things that can happen. Excessive protein can enhance calcium excretion. So remember, calcium is very important for the structure of the bone. So if you lose calcium, your bones will become brittle. Also, if you eat too much protein, it can increase your cholesterol levels. And thirdly, increased protein can create kidney stones. One of the most popular proteins that we know about is a protein called gluten. It is found in grains such as wheat, barley, and rye. The purpose of gluten is it helps the bread have a appealing texture, or it gives elastic properties to the bread. So when we cook bread rises. If you remove gluten, the bread crumbles. So in the image below, you see two types of bread. The one on the right has gluten added. The one on the left does not. The gluten has been removed. So look how fluffy and it has volume on the one on the right versus the one on the left is wrinkly and it looks very sad. So therefore, a lot of people do not have gluten problems. Right now, currently, if you watch the media, everybody is afraid of gluten. But only a few people need to be afraid of gluten. The people who have food allergies need to be mindful of gluten and other molecules like milk sugars and so on. But most of us are just intolerant. So if you're just intolerant to a specific food, you would just have a less severe digestive problem. So you can eat small amounts of it and you will be safe. Versus someone with a food allergy, they might have an allergic reaction where their lips swell, their mouth swells, their esophagus closes, and so on. So those individuals need to be on a strict gluten-free diet. Everybody else can consume gluten. So let's look at what gluten does. When it gets into the digestive system, it travels until it gets to the small intestines. Inside the small intestines, you have these invaginations. These invaginations are called villi. The villi is going to increase the surface area where absorption of the nutrients will occur. Remember, in the small intestines, nutrients are absorbed, and in the large intestines, water is absorbed. So on the villi, you have even smaller projections called microvilli. So all of these are going to be damaged because when gluten gets there, the immune system is going to try to destroy the gluten protein, and it will inadvertently destroy the villi. So therefore, a person with celiac disease, they are allergic to gluten. This gluten will cause their villi and microvilli to be destroyed, and then they will now be malnourished because they cannot absorb nutrients. So gluten is an issue for certain people. This shows how many people have celiac disease. About 2 million people in the United States have celiac disease. That is about 1 in every 133. This website here, amino acids are the building blocks of proteins. We currently have discovered 20 amino acids. These 20 amino acids have a very, very similar structure. They have a centralized carbon called a chiral carbon. Surrounding that chiral carbon, they have an amino group, they have a carboxyl group, and they have a variable group. All of them will have the same structure. The variable group is the only portion that will change. This slide shows several amino acids, and you can see that the red box is the portion that is in common with all amino acids. So the portion in the green is the variable group. That's the portion that changes. So let's look at aspartic acid. That's one of the amino acids that we must consume. The simplest one is called glycine, but aspartic acid has acidic properties because it has a carboxyl group in its variable group. So it has the ability to release hydrogen. So therefore it has acidic properties. Some amino acids are acidic. This occurs because of their variable group. The variable group in aspartic acid and glutamic acid contain a carboxyl group. And remember from previously, we discussed that the carboxyl group can donate a hydrogen. Therefore, it has acidic properties. So if the variable group has a carboxyl group in it, those amino acids would be considered as acidic amino acids or acid-like. Here is an amino acid that has basic properties. Lysine. If you look in its variable group, it has an amino group. The amino group has the ability to receive hydrogen from the solution. Therefore, it has basic properties. And we've talked about cysteine before. Cysteine has a sulfur group, so therefore it can form a disulfide bridge. Some amino acids are basic. These 3 amino acids have basic properties because of their variable group. Their R group has an amino group. Remember from previously, we said that if an amino acid has an amino group, the NH2 can receive an extra hydrogen, therefore pulling hydrogen out of the solution. So if an amino acid has an amino group in its side chain, then it would be a basic amino acid. All charged amino acids, whether they be acidic amino acids or basic amino acids, are hydrophilic. Since they have charges, they would be attracted to water. And if they are attracted to water, they will be orientated on the exterior portion of proteins. Non-polar amino acids tend to be on the interior of a folded protein. Water is going to touch the exterior portion of a protein, but in the interior of the protein, water cannot get there. So all of the non-polar amino acids would tend to be on the interior portion of a protein. These non-polar amino acids are the essential amino acids that you need to consume daily. Polar amino acids tend to be on the exterior portion of a protein. Polar amino acids will have charges. Therefore, it is attracted to water. It is hydrophilic. We're going to look at several proteins now. The first one is hemoglobin. So this is one unit of a hemoglobin molecule. So if you notice, you have the outside portion of this protein and you have the inside portion. The inside portion contains the heme group. In the heme group, this is where iron will bind. And then remember, hemoglobin transports oxygen to the trillions of cells inside of your body. This is another way of looking at the hemoglobin protein. So hemoglobin consists of 4 subunits. If you look at the outermost portion of this hemoglobin protein, those would be where the polar amino acids would be. On the interior of this protein would be where the nonpolar amino acids will be. So proteins are composed of combinations of 20 different amino acids. So if you have at least 10 amino acids, you have a polypeptide. 50 or more, it is called a protein. But most proteins consist of between 100 to 10,000 amino acids. In this image below, this is the primary structural sequence of a protein. The amino acids are linked by covalent bonds that we will call peptide bonds. So now we're going to look at the formation of a dipeptide. We're going to take 2 amino acids and join them together by a dehydration synthesis reaction. Notice that the hydroxyl group of the carboxyl group is going to get together with the hydrogen group of the amino group. This means water will be formed. This water will eventually be released. The water is released in a dehydration synthesis reaction. Once the water is released, a unique covalent bond will be formed. It is called a peptide bond. This creates a dipeptide. A hydrolysis reaction is going to catabolize the dipeptide. It's going to break down the dipeptide. In order to break it down, water will be required. The end product will be the production of 2 monomers. So metabolism is the combination of both a dehydration synthesis reaction and a hydrolysis reaction. Now we will look at 4 structural levels of a protein. The primary level is the linear amino acid sequence. This is when the amino acids are linked covalently, or better yet, through a peptide bond, and they are just held together by that specific bond. The secondary structural level is when that linear sequence winds up like a spiral. So if we look at the image on the bottom, that is a spiral. That is our secondary structure. So imagine a spiraling staircase. Floor 1 is held to floor 2, and 2 is held to 3. So the linear sequence is held by a peptide bond, but each floor in a spiraling staircase is held by hydrogen bonds. So the red arrow shows you what holds one fluor to the next fluor. So in order to hold a secondary structural level together, you need two bonds. The peptide bond originally from the primary sequence, and now an additional bond called the hydrogen bond. And we have two types of secondary structures that can form: an alpha helix and a beta pleated sheet. The tertiary structural level occurs next. We will have that spiraling staircase. Now we're going to take the fifth fluor up to the 10th fluor, break them apart, and fold them down to the first through fifth. This is going to create a globular-like structure that you see over to the right in that image. Well, what holds the first 5 fluors to the next 5 fluors are going to be a disulfide bridge, an ionic bond, and hydrophobic interactions. So now if I ask you what holds the tertiary structural level together, you have to start with the primary and say peptide bonds. Then you have to say that with the secondary, hydrogen bonds. Then you have to say that with the tertiary, hydrophobic interaction, disulfide bridge, and ionic bonds. So all of them collectively will hold the tertiary structure together. The quaternary structural level is when you bring 2 or more of those tertiary levels together. So in the image below, to the far right, we have a globular protein. Each one of those globular proteins in that image is a tertiary structure, and we have 4 of them. We have 2 light purple and 2 dark purple. So when you have all of those collectively together, that is a quaternary structural level. We're now going to talk about 2 types of proteins. One type is fibrous, the other type is globular. Fibrous is going to be strand-like, globular is going to be spherical-like. The lowest functional level of a fibrous protein is the secondary structural level. So it is also functional at tertiary and is also functional at quaternary. These fibrous proteins are strand-like. They are extended. Examples of fibrous proteins are keratin, elastin, collagen, and certain contractile fibers. Keratin is found in hair, nails, and skin. It's also found in feathers and scales. Collagen is found in skin, bones, connective tissue, teeth, gums, etc. Collagen is a helical tropocollagen molecule that is packed side by side. It is the most abundant protein in the body. It gives mechanical support and tensile strength. In this slide, we look at young skin versus old skin. The older skin, the elastin fibers have broken, and the collagen fibers have atrophied. So as you get older, this is what happens when your skin wrinkles. So let's look at the image to the bottom right. This is a slide from your anatomy class. You see what the elastin fibers look like. You don't want those to break. And you see what the collagen fibers look like. You don't want those to disappear or atrophy. Network of elastin and collagen fibers will cause wrinkles. So if your elastin gets broken or your collagen atrophies, it will cause wrinkles. How will that occur? Sun damage. Sun damage can occur to the collagen fibers. It can also cause an excessive production of abnormal elastin. So this can make your skin wrinkle. Also, smoking. Smoking causes reduction of new collagen. So be careful if you sunbathe or smoke. Now we will look at globular proteins. So the functionality of a globular protein occurs in a tertiary or quaternary structural level. The shape of globular proteins are compact and spherical. Examples of such proteins are antibodies, hormones, and enzymes. We will now talk about enzymes. Most enzymes are globular proteins that act as biological catalysts. Some enzymes are holoenzymes, which function only when you have a apoenzyme linked to a cofactor. So in the image below, we show a apoenzyme. This is just the protein portion. This cofactor or coenzyme must bind to the apoenzyme to form a holoenzyme. Once the holoenzyme is formed, basically the cofactor turns it on. Now the holoenzyme can break down its molecule. This picture shows a protein called an enzyme, and it has an active site. The active site is going to be where the substrate binds. Enzymes are frequently named for the type of reaction they catalyze. Lipase is an example of an enzyme. Salivary amylase is another example. So enzymes are named usually because of the substrate they catalyze, and they typically have ASE at the end. The purpose of enzymes are to speed up reactions. So the reaction will naturally occur, but the enzyme will lower the energy of activation, therefore making it more likely to occur. In this slide, we're looking at an enzyme called sucrase. Sucrase will break down sucrose into glucose and fructose. Water is required for this reaction to occur. Here is another example. We have two amino acids are going to bind to the active site. Remember, the active site is like a hand in a glove. The active site will only fit a certain size. So if the amino acids are not the right amino acids, they will not fit in the active site. It's very specific. So the substrate binds, energy is required, water is released, and product is formed. The product in this example is a dipeptide. Now we will look at another type of protein called chaperones or chaperonin. A chaperonin will help other proteins achieve their three-dimensional shape. Chaperonins also help maintain folding integrity of proteins. Chaperonins also help move molecules in and out of the cell. This is called translocation. And fourthly, the molecular chaperonins help to recycle damaged or denatured proteins. This picture depicts what I just described. A unfolded polypeptide comes into the chaperonin, the cap attaches, and what comes out when the cap comes off is a globular or folded protein, correctly folded. We will conclude with talking about how we stain these proteins. We use a compound called biuret reagent. Biuret reagent is nothing more than a combination of two other reagents. We can use potassium hydroxide and copper sulfate, or we can use sodium hydroxide and copper sulfate. When we add those two together in the presence of a protein, we will receive a violet or purple color. That is a positive indicator for the presence of protein. Notice that this is a chemical reaction. Several other tests we looked at are also chemical reactions, versus one test that we looked at is nothing but a stain. This shows you the results that we should receive in lab. Some of them will turn violet and some will not. The ones that turn violet are positive. In this one, we have a positive control.